Glycolysis. So basically, what is glycolysis? Glycolysis is conversion of glucose to pyruvate. In anaerobic conditions, lactate is the end product, and in aerobic conditions, that is in the presence of oxygen, pyruvate is formed at the end. So in aerobic conditions, glucose is converted to pyruvate, and in anaerobic conditions, glucose is converted to lactate. Define glycolysis. Glycolysis is defined as the sequence of reactions converting glucose to pyruvate or lactate with the production of ATP. At the end, synthesis of ATP molecules takes place. Steps in glycolysis. Major three steps. First step is energy investment phase. Second is splitting phase, and third is energy generation phase. As we will see, total 10 steps of glycolysis are there. 1 to 3 steps are energy investment phase. Here ATP molecules are utilized. Second is splitting phase. Step number 4. 6 carbon molecules are 6 carbon molecule is converted to 2 molecules of 3 carbons each. And third is energy generation phase. From step 5 to 10, ATP molecules are produced. First, we will look at the 10 steps of glycolysis, that is conversion of glucose to end pyruvate in aerobic conditions and to lactate in anaerobic conditions. So, first step is conversion of glucose to glucose 6-phosphate. Glucose is converted to glucose 6-phosphate. Glucose 6-phosphate is converted to fructose 6-phosphate. Fructose 6-phosphate is then converted to fructose 1,6-biphosphate. Fructose 1,6-biphosphate is then splitted into 2 molecules of 3 carbons each. 1 molecule of 6 carbon it is converted to 2 molecules of 3 carbons each. First is DHAP, dihydroxyacetone phosphate, and second is glyceraldehyde 3-phosphate. Next, glyceraldehyde 3-phosphate is further converted to 1,3-biphosphoglycerate. 1,3-biphosphoglycerate is formed. Then next 1,3-biphosphoglycerate is then further converted to 3-phosphoglycerate. 3-phosphoglycerate is converted to 2-phosphoglycerate. Just change in the position of phosphate group from 3 to position 2. 2-phosphoglycerate is then converted to phosphoenol pyruvate, PEP, phosphoenol pyruvate. Pyruvate is formed in the next step. Phosphoenol pyruvate is then converted to pyruvate. First in enol form, then enol form is spontaneously converted to keto form. Enol form and then pyruvate keto form. First pyruvate enol form and then pyruvate keto form. This happens in aerobic reactions where oxygen is present. Now in absence of oxygen, that is in anaerobic reactions, pyruvate is then converted to lactate. Lactate is the end product in anaerobic reactions. Now we will look at the enzymes involved. So in the first step, glucose is converted to glucose 6-phosphate with the help of enzyme glucokinase or hexokinase. Glucokinase or hexokinase. Kinase means formation. When there is formation in this step, the word kinase is used. Glucose se glucokinase, and hexo means 6 carbons. 6 carbons, it represents hexokinase or glucokinase. Glucose is converted to glucose 6-phosphate. Now in the next step, fructose 6-phosphate is formed. Glucose 6-phosphate is converted to fructose 6-phosphate. Glucose 6-phosphate, glucose 6-phosphate se fructose 6-phosphate. Glucose and fructose are isomers of each other. So the enzyme named isomerase. Glucose and fructose isomers of each other. So isomerase. Phosphohexose isomerase. Phosphate group. So phosphohexose 6 carbon. Hexose isomerase. Glucose and fructose isomers of each other. Isomerase. Then next, fructose 6-phosphate is converted to fructose 1,6-biphosphate. Fructose 6-phosphate, fructose 1,6-biphosphate. Phosphofructokinase. kinase, phosphate group fructose and kinase, that is the formation. So to remember, we can remember it like this. First step kinase, isomerase, then again kinase. Kinase, isomerase, kinase. Formation, just isomerase, glucose, fructose, isomers, and then again kinase. Now in the next step, 6 carbon molecule. One molecule of 6 carbon is converted to 2 molecules of 3 carbons. Fructose 1,6 biphosphate is split into 2 molecules. One is DHAP and second is glyceraldehyde 3-phosphate. Both 3 molecules, 3 carbons each. So, here the enzyme which takes place is aldolase. Aldolase. DHAP and glyceraldehyde 3-phosphate are again isomers of each other. So the enzyme isomerase. Now phosphotriose isomerase. The whole name of the enzyme is phosphotriose isomerase. Phospho representing the phosphate group. Triose means 3 carbons and isomerase, isomers of each other. Now in the next step, glyceraldehyde 3-phosphate is converted to 1,3 biphosphoglycerate. Glyceraldehyde 3-phosphate to glycerate. 1,3 biphospho. 3-phosphate to 1,3 biphospho. Glyceraldehyde 3-phosphate is converted to 1,3 biphosphoglycerate. Enzyme name dehydrogenase. Then in the next step, 1,3 biphosphoglycerate is converted to 3-phosphoglycerate. 3-phosphoglycerate. Now again here formation of glycerate is taking place. So the enzyme name kinase. Phosphoglycerate kinase. Glycerate is formed. So kinase. Phospho representing the phosphate group. Phosphoglycerate kinase. 1,3 biphosphoglycerate is converted to 3-phosphoglycerate. Now in the next step, 3-phosphoglycerate is converted to 2-phosphoglycerate. Only the position of phosphate group is changing from 3 to 2. So the name mutase, enzyme is named mutase. Same, phosphoglycerate kinase to phosphoglycerate mutase. 3-phosphoglycerate to 2-phosphoglycerate. Now in the next step, 2-phosphoglycerate is converted to phosphoenol pyruvate, PEP. Phosphoenol pyruvate. Enzyme name enolase. So enolase, easy to remember. So 2-phosphoglycerate converted to phosphoenol pyruvate, enolase. Now in the next step, pyruvate is formed in enol form. So formation, so again pyruvate kinase. Then enol is converted spontaneously to its keto form. Enol form is spontaneously converted to keto form. Then pyruvate in keto form is converted to lactate in anaerobic conditions with the help of enzyme lactate dehydrogenase. Pyruvate enol form spontaneously converted to its keto form, and keto form in anaerobic conditions leads to the formation of lactate dehydrogenase. Now we will talk about the reversible and irreversible reactions. So I have already shown it with these arrows. First step, third and the 9th step here, leading to the formation of pyruvate in enol form. First, third and 9th step leading to the formation of pyruvate in enol form are irreversible. First step, third step and 9th step are irreversible. Rest all the reactions are reversible. So I have shown it with this arrow. All the reactions are reversible. Only first, third and the 9th step, that is the formation of pyruvate, are irreversible reactions. Now we will talk about the ADP and ATP synthesis. In the first step and the third step, 2 molecules of ATPs are utilized. 2 molecules of ATPs are used in the formation of glucose 6-phosphate and in the formation of fructose 1,6 biphosphate. 2 molecules of ATPs are utilized. Then 2 molecules of NADH are produced. 2 molecules of NADH are produced here in the formation of 1,3-biphosphoglycerate from glyceraldehyde 3-phosphate. Two molecules of NADH are produced. These molecules of NADH which are produced here in the cycle of glycolysis are further utilized. These NADH molecules are further utilized in the ETC chain. Glyceraldehyde 3-phosphate, two molecules are produced in the formation of 1,3-biphosphoglycerate. Then in the 5th step and in the 8th step, that is the formation of 3-phosphoglycerate and in the formation of pyruvate, two molecules of ATPs each are formed. Synthesis of ATPs, here two molecules of ATPs are produced and here again two molecules of ATPs are produced. Why two molecules of ATPs? Because from here 6 carbon, one molecule of 6 carbon is converted to two molecules of 3 carbons each. So 2 into ATP, 3 carbons into 2. At each step 2 molecules of ATPs are produced. So in the formation of 6th step and in the 9th step, in the formation of the 9th step, total 4 molecules of ATPs are produced. Now we will talk about the anaerobic cycle. In anaerobic cycle, that is in the absence of oxygen, conversion of pyruvate to lactate, two molecules of NADH are consumed. Two molecules of NADH are consumed. Now, as we can say, studies have shown that one NADH molecule leads to the formation of 3 ATP molecules. One NADH molecule leads to the formation of 3 ATP molecules. Now we will look at the energetics, that is the generation of ATP molecules. First for aerobic cycle and then for anaerobic reactions. So first for aerobic reactions, first and third step, as we have seen, two molecules of ATPs are consumed. Two molecules of ATPs were utilized. So minus 2 ATPs. Then 2 NADH. Two molecules of NADH were produced. One molecule of NADH leads to production of 3 ATP molecules. So 2 NADH molecules will lead to production of 6 ATP molecules. So plus 6 ATPs. And in the 6th and the 9th step, 4 molecules of ATPs were produced. Total 2 at 6th step and 2 molecules at the 9th step. So plus 4 molecules. So to brief, plus 6 plus 4 and 2 ATP molecules were consumed. So minus 2. So 6 plus 4 is 10 minus 2. So 8 ATPs are produced at the end of glycolysis cycle for aerobic reactions. In the presence of oxygen, 8 ATP molecules are produced. Now we will look for anaerobic reactions. For anaerobic reactions, same, only the last step, that is the conversion of pyruvate to lactate. Lactate takes place. So at that step, at the 10th step, minus 2 NADH molecules. Minus 2 NADH molecules will lead to minus 6 ATP molecules, because one NADH molecule leads to the formation of 3 ATP molecules. So minus 2 NADH will take place. So minus 6 ATP. So to brief, 6 plus 4 minus 2 and minus 6 ATPs from the 10th step. So that is to brief 10 minus 8, that is 2 ATPs. In the absence of oxygen, that is in anaerobic reactions, only 2 molecules of ATPs will be produced at the end. Now we will look at the structures at each step. Total 10 steps of glycolysis. We will start with the first step, that is glucose, the structure of glucose. As we know, glucose has aldehyde group present. So first is aldehyde C double bond OH, then next COHH, CHOH, COHH, OHH, then last CH2OH. This is the structure of glucose. For glycolysis, we just have to remember the structure of glucose. Rest all the structures are derived from this one structure, that is the structure of glucose. We just have to change one position and the next structure will be obtained. So first we have to memorize the structure of glucose, that is aldehyde group is present. So first aldehyde group, C double bond OH, C OH H, H OH, OH OH this side, and then last CH2OH. Glucose is converted to glucose 6-phosphate. So glucose and glucose 6-phosphate, we just have to add phosphate group at the 6th position, glucose 6-phosphate. Rest, as you can see, both the structures are same. We just have to add phosphate group at the 6th position. Then next, glucose 6-phosphate is converted to fructose 6-phosphate. Glucose 6-phosphate converted to fructose 6-phosphate. Glucose and fructose are isomers of each other. In glucose, we have aldehyde group present. In fructose, we have ketone group present. So, for the structure of fructose 6-phosphate, we just have to add ketone group. First carbon OH, second ketone group. Rest, as you can see, glucose 6-phosphate and fructose 6-phosphate. Same structure. We just have to remember this difference. First glucose, we have aldehyde group. Second, glucose 6-phosphate, we have phosphate group at the 6th position. Then next, glucose 6-phosphate is converted to fructose 6-phosphate. So we just have to change the aldehyde group to ketone group. Then next, fructose 6-phosphate is converted to fructose 1,6-biphosphate. Fructose 1,6-biphosphate. As from the name also we can know, fructose 6-phosphate, fructose 1,6-biphosphate. So at the first position, we have to add phosphate group. First position, phosphate group. CH2OH is converted to CH2OP, phosphate group. Rest, same structure. See, same structure. Fructose 6-phosphate, fructose 1,6-biphosphate. Now, after the formation of fructose 1,6-biphosphate, two molecules of 3 carbons each are formed. Means this one molecule of 6 carbon is splitted into 2 halves. Upper 3 molecules, 3 carbon compounds, as you can see, and here 3 carbons. Fructose 1,6-biphosphate. So I am writing over here. First, CH2 oxygen phosphorus, CH2 oxygen phosphorus, C double bond O, C double bond O, and last CH2OH. DHAP is formed. DHAP is formed. Now the other half. I am writing this upside down. So CH2OP, CH2OP, then next COHH over here, COHH, and last C over here, C double bond O, and here H, aldehyde group. 1, 2 and 3. So to name this, glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. We will take a look at the structure of glycerol. Glycerol is CH2OH, CHOH, and CH2OH. This is the structure of glycerol. Now if we compare the structure of glycerol and glyceraldehyde 3-phosphate. So at the third position we have phosphate group, second we have same, and at the first position aldehyde group is present. So glycerol, name glycerol, then first position aldehyde, so glycerol 1 aldehyde, and at the third position we have phosphate. So glycerol 1 aldehyde 3-phosphate, glyceraldehyde 3-phosphate. Okay. Then next, glyceraldehyde 3-phosphate is further converted to 1,3-biphosphoglycerate. 1,3-biphosphoglycerate. So we just have to add phosphate group at position 1 and 3. Now see this structure. At position 1 and 3, phosphate group. Just add phosphate over here at the first position. Okay. Rest, the structure is same. 1,3-biphosphoglycerate. Now in the next step, 1,3-biphosphoglycerate is converted to 3-phosphoglycerate. 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 So here carboxylic acid group is formed. Rest, the structure is same, as you can see. CH2O phosphorus, CH2O phosphorus, COHH, and here COOH. Once this carboxylic acid group is formed, rest all the structures will have this carboxylic acid group present. So this is the structure of 3-phosphoglycerate. 3-phosphoglycerate. Then next 2-phosphoglycerate. From third position, phosphorus group will move to second position. Just one difference. So this phosphorus has moved to here. as you can see, second position. Okay, 2-phosphoglycerate. Then next we have PEP, structure of PEP, phosphoenol pyruvate. PEP, phosphoenol pyruvate. Phosphoenol pyruvate, so that is the formation of double bond. We just have to remove OH from here and add a double bond. Rest the structure is same. Just add over here double bond. Okay, so this is the structure of PEP, phosphoenol pyruvate. Now for aerobic reactions, last step is the formation of pyruvate. This is the structure of pyruvate. CH3 group, that is a methane, C double bond O and C double OH, carboxylic acid group, and here ketone group is present. Methane, ketone and carboxylic acid. 3 groups. Okay. Now to brief, just remember the structure of glucose. Glucose 6-phosphate, that is phosphorus group at 6th position. Then glucose 6-phosphate to fructose 6-phosphate. So aldehyde group is converted to ketone group. Just one difference. Remember only the differences we have to make in the structure. So first glucose, then glucose 6-phosphate, then fructose 6-phosphate. Just change the group. Then fructose 6-phosphate to fructose 1,6-biphosphate. So we just have to add one phosphate group. 1 and 6 position phosphate group. Okay. Fructose 1,6-biphosphate. Then here splitting. So I have shown it with this dotted line. Splitting. Okay. So 3 carbon and over here these 3 carbons. These 3 carbons will form DHAP. DHAP. And these 3 carbons will form glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Then we can show the structure of glycerol. Glyceraldehyde 3-phosphate is formed. Then next 1,3-biphosphoglycerate. Just add phosphorus. 1 and 3 position will have phosphorus. Just see the difference is what we have to make. So 1,3-biphosphoglycerate. Then next 3-phosphoglycerate. 3-phosphoglycerate. COOH group will be formed. COOH carboxylic acid group is formed. 3-phosphoglycerate. Then next 2-phosphoglycerate. 2-phosphoglycerate. From third position, phosphate group has moved to second position. From third to second position, 3-phosphoglycerate to 2-phosphoglycerate. Then in the next step, PEP, phosphoenol pyruvate. Phosphoenol pyruvate. Enol means double bond. Enol. So here formation of double bond, rest the structure is same. Phosphoenol pyruvate. Phosphoenol pyruvate is then converted to pyruvate. Pyruvate is methane, ketone and carboxylic acid group. 3 groups. We just have to remember these 3 groups. Okay. So that is pyruvate, that is pyruvic acid. So for aerobic reactions, the last step is the formation of pyruvate, and for anaerobic reactions, the last step is the formation of lactate. Pyruvate is further converted to lactate in absence of oxygen. So this is the structure of lactate. COO minus CHOH and last CH3 group, lactate. Okay. So to summarize, glycolysis is conversion of glucose to pyruvate in aerobic reactions, and in absence of oxygen, glucose is converted to lactate. Lactate is the end product. I have provided the notes for this glycolysis in the description below. Hope it will help you. Thank you.